Chapter 5, Section 5. Nucleic acids store, transmit, and help express hereditary information. The amino acid sequence of a polypeptide is programmed by a discrete unit of inheritance known as a gene. A gene consists of DNA, a polymer known as a nucleic acid. Nucleic acids are made of monomers called nucleotides. The two types of nucleic acids are ribonucleic acid, or RNA, and deoxyribonucleic acid, or DNA. RNA and DNA are the molecules that enable living organisms to reproduce their complex components from generation to generation. DNA provides directions for its own replication. DNA also directs RNA synthesis and, through RNA, controls protein synthesis. This entire process is called gene expression. We will cover gene expression later on in a further chapter. The basics of gene expression. DNA remains inside the nucleus. It never leaves because that's where it's safe, can't be damaged, or it's less likely to be damaged. So the synthesis of mRNA, or messenger RNA. So DNA will make a copy of itself, a blueprint copy, in the form of mRNA, which is single-stranded. The mRNA can then leave the nucleus, go find a ribosome, which is made of rRNA. The ribosome will use amino acids that are floating around in the cytoplasm, transfer RNA, or tRNA, to carry those amino acids to the ribosome, where it reads the ticker tape of the mRNA and synthesizes that polypeptide chain. Organisms inherit DNA from their parents. Each DNA molecule is very long, carrying several hundred or more genes. Before a cell reproduces itself by dividing, its DNA is copied. The copies are then passed to the next generation of cells. Although DNA encodes the information that programs all the cell's activities, it is not directly involved in the day-to-day operations of the cell. Each gene along a DNA molecule directs the synthesis of a specific type of RNA called messenger RNA, or mRNA. The mRNA molecule interacts with the cell's protein synthesizing machinery to direct the ordering of amino acids in the polypeptide. The flow of genetic information is DNA to RNA to protein. Protein synthesis occurs on a cellular structure called a ribosome. In eukaryotes, DNA is located in the nucleus, but most ribosomes are in the cytoplasm. mRNA functions as an intermediary, moving those genetic instructions for building proteins from the nucleus to the cytoplasm. Prokaryotes lack a nucleus, but they still use mRNA as an intermediary to carry the message from the DNA located in the nucleoid to the ribosomes. A nucleic acid strand is a polymer of nucleotides. Nucleic acids are polymers made of nucleotide monomers organized as polynucleotides. Each nucleotide consists of three parts: a nitrogen base, a pentose sugar, and one or more phosphate group. The combination of a pentose and a nitrogen base is called a nucleoside. The addition of the phosphate group creates a nucleoside monophosphate, or nucleotide. The nitrogen bases are rings of carbon and nitrogen that come in two types: purines and pyrimidines. The pyrimidines have a single six-membered ring of carbon and nitrogen atoms. There are three different pyrimidines. There's cytosine, thymine, and uracil. Thymine is found only in DNA, and uracil is found only in RNA. Purines have a six-membered ring joined to a five-membered ring. The two purines are adenine and guanine. The pentose joined to the nitrogen base is ribose in the nucleotides of RNA, and deoxyribose in DNA. The only difference between the sugars is the lack of an oxygen atom on carbon 2 in deoxyribose. Because the atoms in both the nitrogen base and the sugar are numbered, the sugar atoms are distinguished by a prime after the number. Thus, the second carbon in the second ring of the sugar is the 2' carbon, and the carbon that sticks up from the ring is the 5' carbon. Here we can see those 5 and 3 ends. You have your 5 and your 3 on that sugar, and this makes up the sugar phosphate backbone. This would be your nitrogen bases. Here you can see the breakdown of what those nitrogen bases look like in a chemical form, and an up-close view of the polynucleotide and nucleotide. components of nucleic acids. We can see the nucleosides. There's the sugars. You can see the numbered carbons. So when we learned that in biochemistry, there was a purpose for that. Polynucleotides are synthesized when adjacent nucleotides are joined by covalent bonds called phosphodiester linkages that form between the OH group on the 3' of one nucleotide and the phosphate on the 5' carbon of the next. This process creates a repeating backbone of sugar phosphate units, with appendages consisting of the nitrogen bases. The two free ends of the polymers are distinct. One end has a phosphate attached to a 5' carbon. This is the 5' end. The other end has a hydroxyl group on a 3 carbon. This is the 3' end. The sequence of bases along a DNA or mRNA polymer is unique for each gene. Because genes are normally hundreds to thousands of nucleotides long, the number of possible base combinations is virtually limitless. The linear order of bases in a gene specifies the order of amino acids, which is the primary structure of our protein, which in turn determines the three-dimensional structure and then the function. Inheritance is based on replication of the DNA double helix. An RNA molecule is usually a single polynucleotide chain. DNA molecules have two polynucleotide strands that spiral around an imaginary axis, forming a double helix. The sugar phosphate backbones of the two polynucleotides are on the outside of the helix. The two backbones run in opposite 5' to 3' directions from each other. This arrangement is referred to as antiparallel. The two polynucleotides or strands are held together by hydrogen bonds between the paired bases. Most DNA molecules have thousands to millions of base pairs. Because of their shapes, only some bases are compatible with each other. Adenine always pairs with thymine, guanine always with cytosine. With these base pairing rules, if we know the sequence of bases on one strand, we would know what the sequence on the opposite strand would be. The two strands are said to be complementary. Prior to cell division, each of the strands serves as a template to order nucleotides in a new complementary strand. This results in two identical copies of the original double-stranded DNA molecule, which are then distributed to the daughter cells. This mechanism ensures that a full set of genetic information is transmitted whenever the cell reproduces. Complementary base pairing can also occur between parts of two RNA molecules or even between two stretches of nucleotides in the same RNA molecule. For example, transfer RNA or tRNA brings amino acids to the ribosome during polypeptide synthesis. The functional shape of tRNA results from base pairing between nucleotides where complementary stretches of one molecule run antiparallel to the other. In RNA, adenine pairs with uracil. Thymine is not present in RNA. Another difference between RNA and DNA is that DNA almost always exists as a double helix, whereas RNA molecules are more variable in shape. Here we can see the differences in the structures between the DNA. You can see the base pairing. The transfer RNA molecule, see how it's paired the same.